Welcome to Med Together. This is a podcast by the people, for the people, as it were. My name is Chana. I'm a med student in New York. This podcast deals with a lot of med school topics, starting from the basic sciences and building its way up, and hopefully it'll be a bit of review that can help y'all consolidate and synthesize important information and maybe phrase it in a way that's easy to understand. By the way, if you're pre-med or you're a nursing student, or you're just interested in sciencey, medically topics, and you're not in healthcare at all, you're totally welcome to. We don't discriminate. If you're interested, you're cool. Ready to rock? Let's do this. Hey everyone, welcome if you're new, welcome back if you're a repeat listener. This talk is in the cell biology series, and today we're going to discuss synaptic transmission at the neuromuscular junction, and then we'll talk a little bit about synapses in the CNS and how they're different and how they're really cool. Okay, so what's the function of a neuromuscular junction? The point here is to transmit an action potential from the nerve to the muscle fiber. This is going to be the prototype for synaptic transmission because it's fairly straightforward, and the only neurotransmitter we use is acetylcholine. It only does one thing. So it's kind of easy to understand as a background, and then we can talk about a little bit more complicated synapses in the CNS. So first of all, just why do we have a synaptic cleft instead of just having the nerve and the muscle fiber touch directly and have the current flow like the way it does down an axon? So here we have an issue of size mismatch. The muscle fiber is very large and you'd need a lot of energy to depolarize it, and the axon just can't carry that much energy. So the signal needs amplification, which is achieved with acetylcholine. And acetylcholine amplifies the signal because every action potential causes 1 to 200 vesicles, each containing 5 to 10,000 molecules of acetylcholine to be released. And they act as ligands on the acetylcholine-gated ion channel, which we call an ionotropic receptor, on the end plate membrane. So how does the presynaptic membrane release acetylcholine vesicles? This is the job of the action potential, basically. This is the whole point. When the action potential in the nerve reaches the presynaptic membrane, which is the end of the nerve, essentially, it opens voltage-gated calcium channels, which allows calcium into the end of the nerve, and calcium mediates the release of acetylcholine vesicles. We're going to elaborate a little bit on the process when we talk about calcium, but basically, calcium binding to surface proteins of the vesicle allows the formation of a fusion pore, and acetylcholine can flow into the cleft. And as we know in biology, oftentimes turning off the signal is as important as turning it on. So we have acetylcholine esterase hanging out in the cleft, and acetylcholine is going to be hydrolyzed fairly quickly. Some of it also diffuses away, and some of it gets reuptaken into the nerve terminal to be recycled. So the structure of the acetylcholine receptor channel. So it's a channel with 5 subunits surrounding a pore. And each of these subunits has 4 membrane spanning alpha helices. You don't have to know this most likely, but just in case you were wondering. And this channel has 2 alpha subunits, and these 2 alpha subunits will each bind an acetylcholine, and they bind the acetylcholine cooperatively, which means that the binding of the first one causes a conformational change that makes the second one more likely to bind. And the selectivity of this channel is such that it wants positive ions and it excludes negative ions because of the residues that line the pore. So, acetylcholine diffuses across the neuromuscular junction, binds to the acetylcholine receptor, which is a ligand-gated nicotinic receptor, and it opens a non-selective cation channel on the muscle fiber. This allows for increased flow of sodium and potassium ions, causing a depolarization of the end plate. The small depolarization that's associated with the release of acetylcholine from a single vesicle is called a miniature end plate potential, MEPP, or a MEP, and it has a magnitude of about 0.4 millivolts. And this actually happens constantly and randomly. The nerve terminals actually just kind of randomly releases acetylcholine vesicles always. So you have it's like blip, 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 every so often. But these are too small. These are mini end plate potentials, right? They're way too small to depolarize anything significantly. But what the action potential does is it causes a ton of mini end plate potentials at once, essentially, by releasing 1 to 200 vesicles at once. And these mini end plate potentials are additive, eventually increasing the end plate potential from resting at about -90 or -100 up to the threshold potential of -60, at which level the voltage-gated ion channels in the postsynaptic membrane open, allowing a flow of sodium ions from the synapse, and you get this sharp spike of depolarization. And this depolarization voltage spike triggers an action potential, which propagates down the postsynaptic membrane, leading to muscle contraction. It's important to note that this end plate potential that we're talking about, this is not an action potential. It triggers an action potential. Now, in a normal muscular contraction, it takes approximately 1 to 200 acetylcholine vesicles, and this causes a depolarization that's 100 times greater in magnitude than a mini end plate potential, which comes from 1 acetylcholine vesicle, right? So if you do the math, this causes the membrane potential to depolarize about 40 millivolts, because 100 times 0.4 millivolts is 40 millivolts. So it goes from about minus 100 to about minus 60, where it reaches the threshold to depolarize the postsynaptic membrane. Now, just a little bit of clarity around this. So you might think instinctively that this non-selective cation channel opens and it allows sodium and potassium through. So sodium's going to flow in, potassium's going to flow out. And you might wonder to yourself, well, how does this cause a change in voltage if you're allowing the same amount of sodium in as you allow potassium out? And here's where you have to remember that at resting, the membrane is much more permeable to potassium than to sodium. So what this channel is doing is essentially leveling the playing field and making the conductance about equal to sodium and potassium. Now, sodium and potassium, since they have similar but opposite intracellular and extracellular concentrations, actually have similar but opposite equilibrium potentials. So in theory, if you allowed this channel to just kind of go forever until it evened out, till it reached its equilibrium potential, you'd end up around zero, because the equilibrium potentials of sodium and potassium would kind of cancel each other out. Now, this does not happen. Like we said, it reaches maybe minus 60, because these acetylcholine channels are open very transiently. They flip open and then they flip shut fairly quickly, because acetylcholine is not binding very tightly to the receptor. It binds, the channel opens, and the acetylcholine kind of pops off. It's a very transient opening, which is why we don't get this huge depolarization. But in theory, I just want you to understand what's happening here. In theory, what's happening is the conductance is equal to sodium and potassium at this point. So if it were allowed to go forever, it would reach about zero, maybe like minus 15, not exactly zero, but somewhere around zero. Anyway, sorry, I digress. To get back to the neuromuscular junction, the pharmacological application of this is that many agents work at the neuromuscular junction to cause paralysis, because if you need this acetylcholine trigger to cause muscle contraction, if you could stop this, you imagine you would get paralysis. So there are three main ways that agents do this. So first of all, they can be competitive blocking agents, essentially, which bind to the acetylcholine receptors and don't allow the acetylcholine to bind. And there's another class of agents which desensitize the receptors, so that even if acetylcholine does bind, it doesn't do anything. This is generally what we use for paralytics that we use in the operating room. And you can also interfere with the cholinesterase and cause there to be usually less cholinesterase. Initially, this causes the response to be sustained for longer, but eventually it's going to desensitize the receptors and cause paralysis of usually breathing muscles. And additionally, hydrolysis of acetylcholine supplies choline to the presynaptic membrane to synthesize more acetylcholine. So if you stop this from happening, the presynaptic membrane is actually going to run out of acetylcholine fairly quickly. And even if you get action potentials, you're not going to be able to release any acetylcholine because there's none left, and this is going to cause paralysis. So some things that do this in a less wholesome way are toxins. So the botulinum toxin, which is the basis of Botox, actually blocks the process of acetylcholine release from the presynaptic membrane. So it interferes with that whole calcium-mediated release, and it doesn't allow acetylcholine to be released at all, which is how it causes paralysis. And actually, tetanus, which is another toxin, does this too initially, but then it undergoes retrograde transport up the axon, and it reaches the neuron in the central nervous system, and it actually disinhibits motor neurons, so there's too much muscle contraction, which is the symptom that we associate with tetanus. And I read a paper recently that discussed the potential of using botulinum toxin to treat tetanus, because they kind of have opposite effects at that point, which is fascinating. I've actually been seeing botulinum toxin pop up a lot recently, used to treat a lot of different neuro stuff, which is very interesting, but again, a digression. I want to mention two diseases also here that really affect the neuromuscular junction. So that's myasthenia gravis is the first one. In myasthenia gravis, there are autoantibodies produced that attack the acetylcholine receptor and cause there to be fewer binding sites for acetylcholine, making it harder to generate action potentials. And contraction leading to muscle weakness, particularly in the extraocular muscles, because extraocular muscles actually are already fairly receptor poor. So if you knock out some of the receptors there, you're going to see an effect much faster. So a typical presentation of myasthenia gravis is going to start with having like a downward gaze because they're not able to really move their eyes, and then it's going to progress to generalized weakness. And then Lambert-Eaton myasthenic syndrome is an autoimmune disorder that affects the presynaptic portion of the neuromuscular junction. This is fairly rare, and it has a unique triad of symptoms. You get proximal muscle weakness, autonomic dysfunction, and areflexia, which is a decrease in reflexes. So proximal muscle weakness is a product of autoantibodies directed against the voltage-gated calcium channels in the presynaptic membrane, which in turn leads to a reduction of acetylcholine release. And then there's also autonomic dysfunction because acetylcholine is a big neurotransmitter in the autonomic nervous system. So this includes erectile dysfunction in men, constipation, dry mouth, dry eyes sometimes, changes in sweating, and areflexia is a condition which tendon reflexes are reduced, and you can imagine why that would be because these are also acetylcholine using. The important association to remember with Lambert-Eaton syndrome is that it often presents alongside small cell lung cancer. Sometimes you can also see Lambert-Eaton with myasthenia gravis. Okay, so the neuromuscular junction, like I said, is fairly straightforward. We're going to run through it one more time. Basically, the action potential arrives at the nerve terminal, opens calcium channels. The calcium allows vesicles of acetylcholine out. The acetylcholine diffuses across the cleft, binds to its receptors, and fairly transiently open this non-selective cation channel and depolarize the end plate enough to trigger an action potential in the next door sarcolemma, which is the membrane of the muscle cell. And that's how action potentials cause muscle contraction. So now let's talk about central synapses a little bit. So central synapses are the synapses we find in the brain and spinal cord, and they're a little different from the neuromuscular junction because the neuromuscular junction is specialized for speed, reliability, and amplification. It's been well studied, it's pretty easy to understand, which is why they're the prototype synapses. Because basically the neuromuscular junction does one big thing. Acetylcholine causes muscle contraction. This is in contrast to the central synapses, which do many different things. It's not just like, 1 + 1 = 2. And this is reflected in their structure and their biochemistry. Brain synapses vary in speed, the kind of transmitters they use, their function. They can be inhibitory or excitatory. And they're specialized for plasticity. They can sum multiple inputs. We have 86 billion neurons, and among these neurons, we have trillions of synapses. And these synapses and the way they work really underlie the functions of our brain and our behavior. We are our synapses in some ways. So again, when we start talking about synapses, we want to talk about why do we need these synapses? Why not direct contact? So unlike in the neuromuscular junction, it's not the size issue, but there are a lot of advantages for having synapses. One of them is that it controls the direction of the information. The information can only flow one way, presynaptic to postsynaptic. If we had a direct touching and electrical transmission, you could imagine that there might be an ability to have retrograde excitation, and that would cause a whole lot of headaches. So this controls the flow of information. It can only go one way. It also allows for more precision in timing, because if you have a chain that has more synapses, it's going to cause the transmission to take a longer time than if you have just a monosynaptic connection. So it can delicately time different events. Also, it allows for inhibition because the activity of one neuron can suppress the activity in another one by acting on a synapse. And it allows for summation. A single neuron can sum inputs from multiple other neurons. So when we talk about summation, there are two different kinds of summation. I just want to mention them briefly, and these can be both inhibitory and excitatory potentials can do summation. And they really have to do summation because these potentials tend to be very small. So summation makes them large enough to produce an action potential in the postsynaptic neuron or to inhibit one in the case of inhibitory neurons. So we have spatial summation, which is summing from multiple neurons, multiple inputs coming in at once to the same neuron. And we have temporal summation, which is rapid firing from a single neuron. So this high frequency input can produce a threshold action potential. So part of what allows for all this complexity is the dendrites, also called the dendritic tree. So this is a network of fibers that receive input and bring it to the cell body. And neurons can have very large, extensive dendritic trees that receive input from many different neurons. Some might be excitatory, some might be inhibitory based on the neurotransmitter they use. And the cell body makes sense of all these different signals. In general, neurons have many dendrites, so many different inputs, but only one axon, only one output. There are exceptions to this because there are always exceptions in biology, but in general, this is a fine way to think about neurons. Many different inputs, one output. So we said neurotransmitters, and we know acetylcholine is the neurotransmitter at the neuromuscular junction, but there are actually a bunch of different kinds of neurotransmitters in the brain. We can have amino acid neurotransmitters, which are like glutamate, glycine, GABA. We can have the biogenic amines, also called monoamines. This is dopamine, serotonin, epinephrine, norepinephrine. These are produced from amino acids. And we can have peptide neurotransmitters. So amino acids are going to be the fastest ones. The biogenic amines are next, and the peptide neurotransmitters are going to be fairly slow. So just another note about neurons. Any given neuron uses only one neurotransmitter. So a neuron is going to be a GABAergic neuron, or a glutamatergic neuron, or a dopaminergic neuron. Like we said, it's kind of similar to the way they only have one axon. That one axon only uses a single neurotransmitter. It can get inputs from lots of different neurotransmitters, but it's only going to act through one neurotransmitter. Some of these neurotransmitters are always excitatory, like glutamate. Some tend to be inhibitory, like glycine and GABA, and others are more diverse. For example, dopamine, it really depends on the postsynaptic receptors. So dopamine can be excitatory to some and and inhibitory to others. And this becomes very important in certain signaling pathways like the basal ganglia. Keep that in the back of your head because that's going to be a long time before we talk about basal ganglia and dopamine, but it is important eventually. So now's probably a good time to talk about the different kinds of receptors. In general, there are ionotropic receptors like the acetylcholine receptor in the neuromuscular junction, which are directly gated by ligands. And then there are metabotropic receptors, which use like a GPCR or a tyrosine kinase. Now, the advantage of direct gating is that it's quick and it's specific. You don't have to invoke any second messengers, which might have other functions. And the actions are going to be very spatially localized. And there's also no ATP needed. So there's no direct energetic component. It's just ions moving down their gradients. And these are going to tend to cause the fastest postsynaptic potentials. Fast postsynaptic potentials are generally mediated by one of the 3 amino acids because they're the fastest. So shortly after the action potential in the presynaptic neuron, you're going to get either a depolarization or a hyperpolarization of the postsynaptic neuron. So glutamate is the most common excitatory postsynaptic potential causing neurotransmitter. What it does is it opens cation channels to get depolarization, versus an inhibitory postsynaptic potential is going to hyperpolarize the postsynaptic membrane. And this is going to be mostly GABA or glycine. And they actually open chloride channels, allowing anions in. And this makes it much harder for the neuron that they're acting on to even act if it would get an excitatory potential because it's so hyperpolarized that it would take way too much to depolarize it to threshold. This direct gating is not that common. It's much more common actually to see neurotransmitters acting via GPCRs, especially the monoamines, but these ionotropic connections definitely do exist. We're going to talk a little bit specifically about the receptors of the amino acids. So GABA and glycine receptors look a lot like the acetylcholine receptor. If you remember, it's composed of 5 subunits surrounding a pore. Each subunit has 4 membranes spanning alpha helices, but the biggest difference is obviously that these are selective for chloride and they exclude cations, so they're going to hyperpolarize the membrane. These are inhibitory neurotransmitters. Glutamate receptors are tetrameric, so they have 4 subunits, and each subunit has a different array. They're actually a little bit of a more complicated family, but there are 2 family members I want to highlight, NMDA and AMPA. So both NMDA and AMPA are ion channels with glutamate receptors. AMPA is pretty straightforward. It binds glutamate, it opens a cation channel. NMDA is the more interesting one and one that becomes very significant. We'll talk about it soon a lot. So it has a glutamate receptor, but it's also bound to glycine. And complicating matters is that there's a binding site for magnesium which closes it. You can think of this magnesium kind of like the sodium inactivation gate. So it has to bind glutamate and not bind magnesium in order to be open. And this happens mostly when the membrane is depolarized. The depolarization of the membrane causes the magnesium to fall off. So it's almost like a second line receptor because there has to be some initial depolarization before there's enough depolarization to knock off the magnesium and activate NMDA. Probably the most significant thing about the NMDA receptors is that they actually allow calcium through, which is not common in these non-selective cation channels. And that's probably what makes it the most important for plasticity, which we will get to very soon. And then glutamate also has metabotropic receptors, which act like any other GPCRs. They just use glutamate as a ligand. Now, the effect of the neurotransmitters really kind of depends on the number and position of the postsynaptic receptors, because if you fire a signal and there are no receptors to receive it, nothing happens, right? So these receptors on the postsynaptic membrane are tightly regulated and controlled, but the number and the position is not fixed, which is important for plasticity. What is plasticity? Plasticity is what allows us to learn and remember things because learning and memory result from specific changes in the transmission capability of a synapse, which is produced by nervous system activity. So basically, the brain can modify its circuitry and make some of it more effective, some of it less effective by processes we call potentiation and depression. So potentiation, usually post-tetanic potentiation, tetanus is a burst of firing, is that after the burst is over, the same action potential is going to produce a larger excitatory postsynaptic potential in that neuron. So basically, you've just upregulated, you've made this neuron more powerful. And then on the other hand, we have something which we call depression, also called habituation, which is that after the firing burst, you might get a smaller response. This could be because of the depletion of available vesicles in the presynaptic terminal. And so sometimes it'll come back after a little while. But if you have a neuron that's receiving slow, repetitive signals, the response is actually eventually going to get smaller and smaller because the nervous system cares most about changes. This makes sense evolutionally. The most important thing is going to be changes. If you're having the same stimulus over and over and over again, it's probably not that important to, your life or death. You want to be able to jump on the stimuli that are changing, which is why the nervous system is most reactive to changes. So these slow, repetitive signals eventually getting less and less response is the biochemical analog of habituation, where if you see the same thing day after day after day after day, you stop noticing it, you stop responding to it. Now, long-term potentiation is basically post-tetanic potentiation that lasts a long time. So this could happen because of convergence of a few tetanic inputs that form a spatial summation, but it permanently shifts the baseline of the neuron. This was first characterized in the hippocampus, talking about memory, but this is a lot of how our learning and memory works. So I want to talk about long-term potentiation with NMDA receptors. So glutamate is going to produce an excitatory postsynaptic potential, initially using the AMPA receptor, because like we said, AMPA is just a simple channel, and the NMDA receptor is voltage dependent because of the magnesium. So a tetanic burst can depolarize the postsynaptic membrane so much that the NMDA receptor is able to open because the magnesium falls off. And calcium comes in through the NMDA receptor channel. And this is what creates a long-term potentiation because it causes growth of new synaptic endings. It creates more places for glutamate to be produced in the presynaptic membrane, and it can even create more AMPA channels. And eventually, this calcium can lead to the building of new dendritic spines and a whole new synapse, which allows for more glutamate receptors, so it's more sensitive to excitation. Long-term potentiation is always going to involve synthesis of new proteins and channels and permanent changes, which make this neuron now more receptive to excitation than it was before. And NMDA is very important in this kind of developmental plasticity, in the activity of psychoactive drugs, and it's even been implicated in epilepsy, even though we don't quite understand how. Now, the same way we can have long-term potentiation, we can also have long-term depression, and this is also NMDA dependent. If a synapse is inactive for too long, the brain suppresses activity from that synapse, and it never really gets back to baseline. It's very, very hard to bring it back, possibly because of AMPA internalization, which makes the postsynaptic neuron less responsive, less likely to turn on an NMDA channel. Now, like we've said before, sometimes ending the signal is as important as starting it. So what do we do with these neurotransmitters when they finish their job? Acetylcholine, like we talked about in the neuromuscular junction, gets hydrolyzed by cholinesterase, and choline gets taken back up into the presynaptic membrane and it gets recycled into more acetylcholine. GABA, glycine, and glutamate can all be recycled directly into the axon terminal, or they can be taken up by glial cells, converted to glutamine, and then brought back to the neuron where they get resynthesized into glutamate, and then glycine and GABA come from there. Serotonin and other biogenic monoamines use a sodium transporter, which loads it back into the presynaptic membrane, and then there are hydrogen co-transporters which pack them back into vesicles. And drugs, both pharmacological and recreational drugs, take advantage of this very often. Think SSRIs for depression. So these selective serotonin reuptake inhibitors block this process, don't allow the reuptake of serotonin. Serotonin hangs around in the synapse longer and can be effective longer. Cocaine does this too, but with dopamine. So whichever example sticks in your head better, cocaine, antidepressants, whatever works. Anyway, okay, that's all for now, folks. We talked about acetylcholine signaling at the neuromuscular junction. We talked a little bit about synaptic signaling and plasticity in the CNS. And I will see you soon in the calcium episode, where we'll talk about how the generation of the action potential at the neuromuscular junction actually causes muscle contraction. Super excited about that one. Calcium might be my favorite topic to talk about. Well, calcium and GPCRs and a few other things, but it's definitely top 10, so get excited for that. As always, reach out to me by email, medtogether26@gmail.com. I'd love to hear from you. And if there are topics you'd like to see covered that you don't see yet, I'm always open to suggestions. Catch you in the next episode.